This is Heejin from Harvard University, and thank you for coming to my talk. Today, I'll be presenting the research that I've done during my PhD, which is about coupling CRISPR scanning with targeted chromatin accessibility profiling using a double-stranded DNA deaminase. So, every somatic cells in the human body contains the same genetic information. However, different cell types serve different purposes. So how does a cell know which genes to express and which to keep inactive to serve its specific role? So this is done through gene regulatory mechanism. Therefore, understanding this gene regulatory mechanism is essential as they play a key role in determining cell identity and in disease development. One way to turn genes on and off has to do with the way that DNA is wrapped around histones to form nucleosomes. So when DNA is tightly bound to histones, the genes cannot be expressed. However, when the genes and the non-coding DNA regions that regulate its expression are accessible, transcription becomes active. And these non-coding DNA regions are called cis-regulator elements, and they include promoters and enhancers. Cis-regulated elements regulate transcription by recruiting transcription factors and other regulatory proteins, which help initiate transcription by RNA polymerase II. Therefore, cis-regulated elements' activity is linked to chromatin accessibility, which is determined by nucleosome positioning and transcription factor binding. In general, so closed chromatin is associated with gene repression, and open chromatin is associated with active transcription. Also, mutation in cis-regulatory elements can change chromatin accessibility and gene expression, which could lead to disease and phenotypic variation. To understand the effect of genetic variants on cis-regulatory elements, we can use CRISPR-based genome editing tools. The CRISPR-Cas9 system uses a single guide RNA to direct the Cas9 protein to a specific DNA sequence where it introduces a double-stranded break. This break is typically repaired in a way that introduces insertions or deletions. Alternatively, we can also use base editors, which consist of a deaminase fused to a nicking Cas9 to introduce precise point mutations without making double-stranded breaks. So using this CRISPR-Cas9 or base editor, we can perform CRISPR mutational scanning on targeted cis-regulatory elements by using guide RNA library tiling across the target region. So after CRISPR mutagenesis, each edited cell contains different mutations within the target CRE, and some of them may impact gene expression and result in phenotypic changes. And next, cells are sorted based on a phenotypic readout, and guide RNAs can be sequenced from each sorted cell population. So from this data, we can link specific sequence perturbations on the targeted CRE to their functional effects. For example, the Dan Bauer group performed a CRISPR-Cas9 screen of the BCL11A enhancer, demonstrating that targeting this enhancer increases fetal hemoglobin expression. Because increasing fetal hemoglobin expression is a key therapeutic strategy for treating sickle cell disease, this development led to the development of the first FDA-approved CRISPR-Cas9 therapy for the sickle cell disease. However, the readout of such typical CRISPR mutational scanning is limited to phenotypic outcomes, such as fetal globin levels in this case. So as a result, the CRISPR mutational scanning does not capture how genetic perturbations affect chromatin structure, even though this is critical for understanding the underlying molecular mechanisms. In addition, this approach identifies the guide RNA sequence, but not the endogenous edited genotypes. Since each guide RNA can generate different editing outcomes, and even low-efficiency guide RNA may not produce any edits at all, it becomes important to directly read the endogenous genotypes to accurately understand the effects of each perturbation. Therefore, to address these limitations, my research focused on developing a new method that couples CRISPR mutational scanning with chromatin accessibility profiling at targeted loci. So the goal is to achieve the following 4 key points. First, I would like to simultaneously detect CRISPR perturbations and chromatin accessibility changes. Second, instead of simply reading the guide RNA sequences, we want to directly detect the resulting edited genotypes. Third, the method should be scalable for large-scale CRISPR mutational screens, which are capable of profiling hundreds of genotypes in a high-throughput manner. Finally, the approach should be applicable to primary cells, which often have limited cell numbers. So there are already many methods that can measure chromatin accessibility, but capturing both accessibility and CRISPR edits require long resequencing, which means that I want to detect chromatin accessibility across multi-kilobase single DNA molecules. One such method is FiberSeq, which uses an M6A methyltransferase, which selectively methylates adenines in open chromatin. And these M6A marks are directly detected using long resequencing. However, FiberSeq has a key limitation. These M6A marks are lost after PCR, so that the library cannot be amplified by PCR. Therefore, FiberSeq requires large amounts of starting material and deep sequencing to achieve sufficient coverage per locus. To address this limitation, we use base editor to map chromatin accessibility. This is possible by using an enzyme called DDDA, which is the double-stranded DNA cytidine deaminase. So unlike other single-stranded DNA cytidine deaminases, mostly used in typical base editors, DDDA can catalyze cytidine deamination within double-stranded DNA. And while the wild-type DDDA is known to have a strong sequence preference on TC context, in this study, to increase the resolution, we used one of the DDD variants, DDD11, which has reduced sequence bias compared to the wild-type DDDA. Therefore, our goal is to use DDD11 as a biochemical tool to profile chromatin accessibility. So adding DDD11 to permeabilized cells will generate genome-wide mutations, but the mutation rate of C2U deamination depends on the degree of DNA accessibility. So DNA regions that is bound by transcription factors or nucleosomes are protected and get less mutated, which leads to the mutational footprints. So the first thing that I've done is to map the distribution of cytidine deamination across whole genome. I treated permeabilized cells with purified DDD11 and performed whole genome sequencing. And you can see that cytidine deamination signals were enriched at CTCF binding sites and transcription start sites, which are known to be open chromatin. Also, it shows the nucleosome phasing patterns around these regions, showing that nucleosomes are binding around this center. Therefore, these results demonstrate the ability of DDD11 to mark accessibility chromatin by mutational profiling. Since chromatin accessibility profiling with DDD11 does not involve DNA cleavage, it is compatible with targeted PCR amplification and long-range sequencing. Therefore, if you're interested in chromatin accessibility at specific loci, instead of performing expensive whole genome sequencing, we can perform targeted PCR on DDD-treated genomic DNA and sequence the amplicons using long-read sequencing. So we named this technique as targeted deaminase accessible chromatin sequencing, TDAC-seq for short. So TDAXI can enrich specific genomic loci of interest with high coverage, achieving over 1,000-fold coverage per region, which was not possible with previous long-read sequencing-based techniques. So this is an example of TDAXI applied to two different cell types. After extensive optimization, I was able to increase the mutational rate high enough to visualize chromatin accessibility at the single molecule and single nucleotide level. So TDAXIC signals aligns well with the ATAC-seq, which is like a typical way to measure chromatin accessibility using short-read sequencing. And you can see that it shows the cell-specific accessibility. So the blue plot shown here indicates the C2T edits, and the red plot here shows G2A edits, enabling a strand-specific view of chromatin accessibility. Also, from the TDAC-seq, we could sequence amplicons up to about 11 kb using nanopore sequencing. Because TDAC-seq profiles chromatin accessibility across long single DNA molecules, we next asked whether we could reveal correlations between two distal accessible sites. To do so, first we defined the deaminase accessible DNAs, which we call as DADs for short, as continuous stretches of DDD11 mutations that mark open chromatin regions on individual DNA molecules. So you can see that the DAD position aligns well with the aggregate TDAC-seq and ATAC-seq signals. And from those DADs, we observed that nearby distal TDAC-seq accessible sites often appear open together on the same DNA molecules. So this highlights the power of using long-read sequencing to detect the co-accessibility between distal accessible sites. Additionally, TDAC-seq can detect protein binding through mutational footprints. To test this, we parked the dCas9 at the target region using guide RNA and compared to a condition without the guide. So you can see that there's a depletion inside the open region when dCas9 protein is sitting in the DNA and protect that DNA sequence from getting mutation. So this demonstrates that TDAC-seq's ability to detect protein occupancy. Next, we wanted to use TDAC-seq to detect endogenous protein binding as well, such as nucleosomes and transcription factors. And to more quantitatively measure the mutational footprints, we calculated footprint scores by adapting the ATAC-seq footprint calling methods. So the way that it is performed is we first define a window consisting of a center region and two flanking regions. And we slide this window across the genome, and if the center region has a significantly lower mutation rate due to protein binding compared to the flanking region, this leads to the high mutational footprint score. Also, to consider the DDD intrinsic sequence bias, we did the bias correction to remove this artificial factor by using the naked DNA data. So by using the I used the DDD whole genome sequencing data and observe the aggregate nucleosome and transcription factor footprints. And we saw the regions that are centered around CTCF or YY1 binding site. And also, one thing that's very useful about this technique is by changing the footprint window size, we can detect the different size of protein footprint separately. So for example, if I set the footprint window as 20 base pair, I could detect the CTCF footprints. However, if I increase the footprint window up to like 50 to 70 base pair, I was able to detect the nucleosome footprints. So also depending on the transcription factor types, it shows a different intensity of TF footprints as well as different nucleosome factor patterns. So compared to YY1, you can see that CTCF footprint is much stronger And also it shows more symmetric nucleosome phasing pattern compared to YY1, which shows like asymmetric phasing pattern. So the previous plot shows aggregate DDD footprints, and I also code mutational footprint in a single DNA molecules from Tdaxic data. The purple plots here represent Tdaxic raw signal detected as C2T or G2A mutations. And the green plot here indicates the nucleosome footprints using a 50 base pair footprint window. And you can see that nucleosome footprint aligns well with MNA-seq, which is a typical way to measure nucleosome binding, but using a short resequencing. And the lower green plots show TF footprints by using a 25 base pair as a footprint window, and it shows strong CTCF footprints right under the CTCF motif. So far, I've shown that we can use Tdaxic to detect chromatin accessibility and protein binding in single DNA molecules. Next, I want to combine Tdaxic with CRISPR perturbation targeting cis regulatory elements. So, after introducing CRISPR mutations, I perform Tdaxic, which introduces DDD mutations on the top of the CRISPR mutated DNA. Therefore, we can detect chromatin accessibility and CRISPR edits simultaneously from a single DNA molecule and see how the CRISPR perturbation affects the chromatin accessibility. So to test this concept, I first disrupted one of the CTCF binding motif in Hoxsey locus using CRISPR-Cas9 and performed Tdaxic on this region. So from the sequencing data, we deconvolute the reads without CRISPR deletions versus reads with CRISPR deletions around the guide RNA binding site. So the plots on the left are from reads without CRISPR deletions, and plots on the right are from reads with CRISPR deletions. And again, each row indicates raw Tdaxic signal, nucleosome footprints, and TF footprints. So you can clearly see that when there's a CRISPR deletions around the guide RNA binding site, you can see the chromatin accessibility decreased compared to the wild type reads. And also this caused the weaker signal of nucleosome and transcription factor footprints. This is because CTCF could no longer bind to the locus after deletion of this motif, and this altered the chromatin accessibility and protein binding of this region. So here I want to emphasize that both datasets comes from a single experiment, demonstrating that we can just computationally separate reads based on the CRISPR edits. Next, I wanted to see if I could use Tdaxic on primary cells as well. And the system that I tested was chromatin accessibility changes during erythroid differentiation of CD34 HSPCs. So during the erythroid differentiation, we know that cells passes different phases, and during the process, HBB promoter becomes more open and HBD promoter becomes more closed. So I perform Tdaxic at 3 time points during erythroid differentiation, and this Tdaxic spanned both gene promoters and it captured the expected changes. So you can see that across those 3 time points, HBB becomes more open and HBD becomes more closed. So this indicates that our method can be also applied to primary cells. Also, in this beta globin region, there's also HBG1 and 2 gene, which control the fetal globin expression. Although these genes are typically silenced in adult blood, reactivation of fetal globin is a way to treat sickle cell disease. So that's why scientists have been very interested, was trying to manipulate this region to treat sickle cell disease. And to reactivate this fetal globin expression, Novartis have used CRISPR-Cas9 therapies using guide RNA 68. One interesting feature about this locus is that HBG1 and HBG2 shares a very similar sequence. So if you cut the promoter using one single guide RNA 68, it targets the both promoter and results this 5kb deletion. And this deletion results in about a hybrid fusion where the HBG2 promoter becomes fused to HBG1 gene body. And this large deletion leads to increase in fetal globin expression and had been also applied to human patients with sickle cell disease. However, the exact mechanism behind this reactivation remains unclear. So to investigate chromatin accessibility in this region, we conducted TDAC-seq after cutting the region with CRISPR-Cas9 using guide RNA 68. As expected, we observed about 5kb deletions after Cas9 cutting compared to the non-targeting guide RNA, and this led to the upregulation of fetal globin expression. So by using these cells, I analyzed this locus through both ATAC-seq and TDAC-seq. So this is like ATAC-seq data, and because ATAC-seq is short-read sequencing, it could not detect large deletions or distinguish between HBG1 and HBG2. As a result, I observed both HBG1 and HBG2 accessibility increase after the edits, even though we already know that HBG2 gene body was removed in most of the cells. So this ATAC-seq data seems to be inaccurate, and it is not an appropriate method for studying this system. However, TDAC-seq could determine the exact genotypes, and all genotypes with this 5kb deletion showed increased accessibility at the HBG1 promoter. And these experiments demonstrate that TDAC-seq can help study perturbation effects of multi-kilobase scale of genomic deletions in homologous regions. So far, I've shown that TDAC-seq coupled with CRISPR perturbation using a single guide. And our next application was to integrate TDAC-seq with pooled CRISPR mutational scanning. So first, cells were transduced with pooled guide RNA library tiling the target cis regulator elements. Next, either Cas9 or ABE base editor was electroporated to generate a pool of mutant cells, each containing different CRISPR perturbation. And then I performed TDAC-seq on this cell population to profile how these different edits affected chromatin accessibility at the targeted region in a high throughput manner. And the first cis regulator elements we chose for CRISPR screening is HS2, which is one of the enhancers that control globin gene expression. First, I conducted the screen using Cas9 cutting and using 21 guide RNA library, and this generated 541 unique deletion genotypes, each with more than 400 coverage. And as expected, the deletions were enriched around the guide RNA binding site. So this is the plot that shows TDAC-seq results across all 541 genotypes. So each row represents different genotypes with purple color indicating TDAC-seq signal intensity. So dark purple corresponds to high chromatin accessibility, while the white indicates close chromatin accessibility. And the small black lines inside each row indicates CRISPR deletions for each genotype. And the right plot shows it's like a different visualization of the same data, and it shows the difference in TDAC-seq signals compared to wild-type reads. So blue color indicates decrease in accessibility, while the red indicates increase in accessibility compared to the wild-type reads. And you can see that there is a very strong blue colors in the middle, especially when it's especially close to the guide RNA 10 and 11 deletion. And so that it shows the most significant effects in reducing chromatin in reducing chromatin accessibility. So next, we wanted to zoom in this part, and we did motif clustering analysis to find the minimum motifs whose deletion altered the chromatin accessibility. And we found that this DNA reads where this 10 base pair key motif is entirely deleted has significantly reduced chromatin accessibility at HS2 compared to other reads. And this key 10 base pair motif mostly overlap with AP1 motif, which is known to be essential for HS2 activity. So this suggests that AP1 is an important TF for making this enhancer accessible. Next, to validate this screen result, I performed a single guide validation using a TAC-seq, and the guide that we chose is guide RN11, which is close to the most to the blue big chunk that showed a significant reduction in chromatin accessibility, and guide RNA1 and non-targeting guide RNA as a negative control. So again, like guide RNA11 targets that 10 base pair key motif, while guide RNA1 targets a site far from the key motifs. So I conducted the TAC-seq following individual guide transduction and Cas9 electroporation. And from the TAC-seq data, I confirmed that guide RNA11 reduces the HS2 accessibility, which validated our TAC-seq results. And this reduction in accessibility was also followed by decrease in globin expression, as shown by RT-qPCR assay. So the previous screen used Cas9 cutting, and next here we repeated the same experiment but performing the ABE base editing screen on HS2. And using the same guide RNA library, here this ABE screen generated 49 unique genotypes. Because TDAXI can directly detect endogenous edits, we were able to group genotypes based on the number of ABE edits. And notably, you can see that TDAXI reads with multiple ABE edits show a greater reduction in HS2 accessibility compared to those with a single ABE edits. And this shows the cooperative effects between ABE edits. And here I took a closer look at the top genotype showing the strongest effect, which include the perturbation of AP1 motif and FOXK2 motif. The fact that AP1 motif emerged again as one of the top hits is consistent with our previous Cas9 screen. In addition, if you look at the exact genotypes, you can see that those with multiple edits, multiple ABE edits, again show like stronger effects on chromatin accessibility than the ones with just a single ABE edit. So the final experiment that I performed was TDAXI coupled with a large-scale CRISPR-Cas9 using more than 100 guides in primary cells. As the target region, we selected one of the GFRB enhancer, which harbored the MPN-associated risk variant in CD34 HSPCs. And previous studies from Sankaran lab have shown that deleting the entire enhancer reduces GFRB expression. However, the exact identity and position of functional sites within this enhancer is still unknown. However, one challenge in studying this enhancer using TDAXI was that GFRB enhancer is relatively close, making it difficult to detect accessibility with DDT11 due to its low mutation rate. Fortunately, however, last year, NAB discovered several new double-stranded DNA cytidine deaminases from different bacterial strains, and some of these newly identified enzymes showed much stronger activity than the wild-type DDDA. So I purified one of the most active enzyme, I'll refer to as MGY enzyme, and I use it for TDAXI. Another key advantage of this enzyme is that it does not have any sequence bias, meaning that it can mutate any cytidines regardless of its sequence context. So when I performed TDAC using this new MGY enzyme, it dramatically improved efficiency and enabled us to successfully profile chromatin accessibility at this GFRMB enhancer. Therefore, using this MGY enzyme, I conducted TDAC combined with CRISPR-Cas9 scanning targeting the GFRMB enhancer and CD34 HSPCs. In this screen, we used more than 100 guides, and this generated nearly 1,000 distinct genotypes. So here, I'm showing the TDAC results across all 947 genotypes, and just from the glimpse, the results seems to be more complex compared to our previous HS2 screen. So to better understand the key motifs within this enhancer, here is an alternative visualization of the GFRMB enhancer screen. So each dot represents a distinct genotype, with the x-axis showing the start position of the deletion and the y-axis indicates the length of the deletion. And the color reflects the change in TDAC signal relative to wild-type reads, like I showed before, while the size of the dot represents the statistical significance of that change. And here we identified 4 key perturbations that significantly reduce accessibility. And through the same motif clustering analysis, we further identified the minimum motifs whose deletion altered chromatin accessibility. And we found that deletion of GATA, AP1, and SPE1 motifs significantly reduce accessibility. And notably, GATA and SPE1 are critical transcription factors in early hematopoiesis, which is the cellular state of CD34 HSPCs. So altogether, these results demonstrate that TDAC enables large-scale functional dissection of key enhancer elements in primary cells. And I want to emphasize that the scale and the resolution of data shown here are uniquely enabled by TDAC due to its compatibility with PCR and not attainable with other long-read sequencing methods. So to summarize this talk, we developed TDAC, which maps DNA accessibility as single molecule, single nucleotide resolution. And coupling TDAC with CRISPR perturbation enables simultaneous detection of accessibility and various types of CRISPR edits on the same DNA molecule. Unlike ATAC-seq, TDAC could detect regained accessibility at HBG1 and 2 region after large deletions. And lastly, TDAC coupled with pooled CRISPR scanning enables massively parallel profiling of chromatin accessibility across hundreds of genotypes. And this research has been published recently on Nature Methods about 2 months ago. So please check it out if you're interested in, and let me know if you have any questions. And so thank you for listening to my talk, and I would like to thank my PhD lab, especially my PI Brian, and all the past and current members of the Liao lab, as well as our amazing collaborators from Brewster Lab, Blue Lab, and Sun Crown Lab. And I would be happy to take any questions.